presenting results yet. We are presenting a project and we are looking for collaborators. So I'm going to be talking about the in silico screening for universal and personalized drug regimens with geroprotective properties. I'm also the director and trustee of the Biogerontology Research Foundation. It's a charity in the UK which acts as a think tank and supports some basic research. So a short story of today's talk. You might get slide 6, so if you want to know the story right away, you can just take a look at this slide and then forget all the others. So what we did at Silico Medicine is that we developed a platform-agnostic algorithm for comparing gene expression data coming from a variety of microarray platforms and NGS. So that allowed us to tap into legacy databases, legacy gene expression databases, and be able to develop a signaling pathway analysis tool that uses signaling pathways, combinations of signaling pathways, as biomarkers of cancer. Then we actually went into personalized medicine of cancer. I'm going to briefly touch on that. And we found ways to manipulate the signaling pathway activation profiles in silico with a variety of drugs with known molecular targets. And moved into personalized medicine and oncology, so it's a running business. And then we expanded into age-related diseases, so we took an already established sets of algorithms and decided to apply them to aging. And now we're moving into geroprotective screening and geroprotector combinations. So we've got a lot of publications this year. This is just a few of them recently. I'm going to briefly touch base on some of them. And you're probably aware of this company, Ingenuity. Some of you are definitely using their software. They were acquired by QIAGEN by about 120 million last year, or this year. And they provide pretty sophisticated pathway analysis tools, where you actually also can compare functionally, like quantitatively, signaling pathway activation states between two different profiles. But I think we can do it better. You know the company called Jingo. They are also used for pathway analysis in a variety of academic institutions and many publications. So they were acquired by Thomson Reuters 2 years ago. Radiant Genomics, also a gene expression data analysis tool. They were acquired by Elsevier. And what's common about those two companies is that their R&D offices were located in Russia, and after they got acquired, they kind of moved into other directions and maybe also other countries. So we hired a few of their people and decided to develop a signaling pathway analysis tool for clinical applications. And we asked ourselves a question whether we can actually do pathway analysis better than the others, so to tap into legacy databases and to be able to derive some clinical value from the pathway analysis, pathway activation analysis, and whether we can group the elements in the pathways differently than everybody else and use them as biomarkers of cancer. So one of the top problems with using gene expression, when using gene expression data, is the high cross-platform variability. There are lots of high error rates, and it's very difficult to compare RNA-seq versus microarray data, even if you take data from, if you take biopsies from the same biological sample and run them on different platforms, let's say 2 different metrics devices, you are going to have a lot of cross-platform variability, and we needed to suppress that. Of course, there is lots of legacy databases to tap into. You can, if you find a way to intelligently compare them, you can tap into almost 500,000 transcriptomes, if you like, with some of them with really good data and good publications attached to them. So we published at the beginning, we published in March, an algorithm that describes how we measure intracellular signaling pathway activation strength and profiles in cancer, and call this algorithm the OncoFinder. For those of you interested in math, I was told to skip it. You can go in and look at the functional kinetic model that we've developed for analyzing the signaling pathway activation profiles, and also evaluating the weight of every element in a network. And we've had 13 people working for 1.5 years to annotate every single element in the pathway with its activator or repressor role to be able to put it into the formulas. We published a paper in Nature Leukemia, so that was our group. I wasn't one of the co-authors, didn't actually contribute. And here what's interesting about this study is that we managed to use the signaling pathway activation profiles to narrow down to individual genes, to individual gene expression, and also identify the optimal way to treat a specific population group. So we can go back to this publication. Afterward, we actually showed that we can use the OncoFinder algorithm to minimize the errors introduced by high-throughput methods of transcriptome analysis, and essentially be able to tap into the legacy databases. And we showed that for the same biologic samples analyzed on different platforms, so both microarray and NGS, we can get fairly good correlations, much better than when we're looking at the log of case to norm for individual genes. We have much stronger correlations when we're looking at pathway activation strength for microarray data versus NGS. And of course, if you group them, you can see that there are much higher correlations between replications of the experiment than when you are looking at the logarithmic case to norm for individual genes. We published another strong paper in OncoTarget. It's a hybrid journal in oncology. I think it's 6x6 factor. And we showed that signaling pathway activation profiles make better markers of cancer than expression of individual genes. And here, what we did, we took a very large number of samples from a number of cancers and looked at area under the curve for identifying that particular cancer using both logarithmic case norm and using signaling pathway activation profiles. And so AUC1 was when we did it through signaling pathways. AUC2 was when we did it through differential gene expression analysis. And you can see that in most cases, signaling pathways were better markers of cancer than expression of individual genes. They had higher area on the curve, over 75. I'm going to briefly skip through that. I'm just showing that we had some interesting background before we started going to cancer and aging. So, and there is a big problem in clinical oncology, which is there are a lot of drugs that work very well, but only in a very limited number of patients with a specific cancer. And if you are reading Lancet Oncology, before going to bed, you see that papers that describe a clinical trial show that no patients showed complete response, but let's say one arm with a specific mutation, so 8% of the patients showed some response, and another arm showed just slightly better response, another 20%. So every time the doctor gives a targeted drug to a patient with cancer, he's rolling a dice. Well, not every time, but most of the time. And wasting precious time and, very importantly, money. So what we try to do, we try to repurpose our software to personalized medicine in cancer, and developed a protocol where we take the tumor biopsy or a paraffin block. So our algorithms are good enough to be able to compare gene expression data coming from both biopsies, fresh biopsies, and paraffin blocks. Extract gene expression data using either custom array system or NGS. Then we do pathway activation analysis, map gene expression data onto signaling pathways, and then we calculate the drug score for every individual drug that's in the database, which is either approved for that particular cancer or is going through clinical trials. We've founded a company called Pathway Pharmaceuticals in Hong Kong for cancer. Decided to separate the personalized medicine and drug discovery initiatives, because personalized medicine is also for cancer, it may be a source of liability. And you probably know a company, Foundation Medicine in the US, so they are providing recourse to doctors and patients based on their tumor profile. But we decided not to go into the US and started operating in China and Russia, and developed a software called the OncoFinder. I'm not going to go into that, but essentially it allows you to play around with pathways, look at what's activated, what's downregulated in cancer versus norm, and get a drug score for every individual drug approved for a specific condition. So essentially to prioritize the drugs. Then we did in silico validation, internal clinical trials, ran through over 250 patients and established research collaborations with top oncologists, and also started working with a company out of Johns Hopkins, which works on mice, so essentially putting patient tumor grafts in mice and profiling the tumors before and after treatment with a variety of drugs. So we are rerunning this data using our software, going to be a publication quite soon. So we started in cancer before going into aging, because all cells try to survive, but cancer cells try harder. And we need to understand the interplay of signaling pathways in cancer and aging on the tissue-specific level. And we used very similar algorithms as we use in oncology for geroprotector screening, and then we'll launch the statistical module to track tissue-specific pathway activation states in healthy patients, cancer patients, and age-related diseases. So also Parkinson's and Alzheimer's, to see what common pathways get activated in cancer, aging, and age-related diseases. So we are tracking this right now. And we founded a company called Silico Medicine in Baltimore, United States. And we also use a very similar pathway cloud when we are analyzing either data from young patient cells and old patient cells, or you can basically look at any cell state or tissue state through the prism of pathway activation profiles. And this is basically, we're looking at just top several pathways that go through a specific threshold, go over the specific threshold to map it and visualize it, but we are looking at almost 300 pathways. And our goal is to find drugs that minimize the signaling pathway disturbance. But not for all pathways, only for pathways that we consider to be pathologic. Because as we age, cells start feeling uncomfortable and activate the defensive pathways. And when we inhibit those pathways, cells die because of the accumulated damage. So we shouldn't really touch them, those pathways. We really need to focus on pathologic pathways. And this is our major work right now. So one of the posters that we presented at MBO, FEPS, showed that, sorry, it's not visible very well, but essentially it shows that we took fibroblasts that went through 50 passages, 70 passages, and 80 passages, and we show that using signaling pathway activation profiles, we can clusterize the pathways very well. So you can visually see that there is a drift in replicative aging, and they clusterize very well by a number of passages. So we developed an algorithm for in silico geroprotective screening, where we take transcriptome data from young and old patients, map the gene expression data onto the signaling pathways, implicated in aging processes, mostly pathologic pathways. Then we construct the pathway cloud, and using a database of drugs with known molecular targets, and right now there are lots, so broad data actually is at least 1,400 drugs that have the effects on transcriptomes in various cell lines. And from literature, we assembled almost 16,000 drugs with known effects on molecular targets, and called it Pharma Atlas. And we calculate the geroprotective rating for each individual drug to evaluate the signaling pathway disturbance effect and the effect to minimize the signaling pathway disturbance for every drug. Afterward, we test the prediction in vitro in human cells. So currently, several rounds of experiments went through in human fibroblasts. First rounds failed, and I'll tell you why. And also confirm the predictions of in silico screening using nematodes. And if rating is confirmed, we increase the number of drugs. So for the first part, we used 250 drugs that could act as geroprotectors. So actually, rapamycin scored very well. And if the rating is not confirmed in vitro and in vivo, we adjust the algorithms to see what's wrong. We published a paper on that in Frontiers. You can take a look. And we developed an internal software system that has a very large number of drugs, so 16,000 drugs, maybe 2,000 drugs with very good, precise effects on gene expression data in a variety of cells, in a variety of tissues. Large transcriptome database, genetics database, so methylation, microRNA, and we have several databases of pathways that are implicated in aging and various age-related diseases. And our approach to geroprotector selection is based on several notions. So one is that it has to be personalized. We age at different rates, and different systems age at different rates. So we actually need to target the regimen to a specific patient and also to a specific tissue. And if we are to find universal geroprotectors, they need to act very well on pathologic pathways in the brain tissue, muscle, and stem cells. Most of the other tissues we consider as being renewable, and maybe it even makes sense to clear the senescent cells and tissue with faster recycling and inducing this clearance. So, for example, one of the experiments that failed so far in fibroblasts, after we predicted the geroprotective rating for 250 drugs and took top 10, tested them on fibroblasts, and I think for 6 of the drugs, fibroblasts died. One of the reasons, so on old fibroblasts, and one of the reasons we believe that they died, just looking at the drugs and how they act, is because we try to also minimize the signaling pathway activation in the defensive pathways. So we essentially try to minimize everything. And we shouldn't do that because that leads to cell death because of the damage that's already accumulated will prevail and kill the cell. In many tissues, that's actually beneficial because it clears out the senescent cells. And also, we need to understand the intervals with which we are going to take drugs. So either we're going to take them continuously or try to induce the chromatic effect, repair the damage, induce the endogenous repair mechanisms, and then have a period of non-taking a drug. So, and the drugs need to work in concert so that the benefit to the patient is maximized while the adverse effects are minimized. So for combinations, and this is currently in process, so we developed the algorithms, we filed for a variety of patents on that as well, because we are trying to do global good, but our investors want IP, unfortunately. We developed several databases. One is the drug database, so again 16,000 drugs. For many cell types, we have transcriptome effects before and after, toxicity data, adverse effects, keystone norm tables calculated from the drug transcriptome effects, and pathway activation strength effects. And we have the gene expression database, which has almost every openly available gene expression data set in existence, plus we license some data. And for every of the combination where we can compare norm to disease and young to old, we calculate the pathway activation drift between healthy norm or young samples and disease or old samples. And our idea is to take the healthy norm or young patients' cell tissue and transcriptomes, cellular tissue transcriptomes, compare the pathway activation strength with the old patients' cell and tissue transcriptomes. Then identify what pathways are pathologic, what pathways are defensive, and try to find drug combinations for drugs with known effects on pathway activation strength so that the effect is maximized and the toxicity is minimized. But again, that should work on a tissue-specific level, and we're targeting brain, muscle, and stem cells as the first priority. And of course, in some cases, we need to think about the person's age and person's risk profile. In some cases, we also calculate the net toxicity and adverse effect index. And in some cases, when the patient is 70 and has predisposition to a variety of diseases, and maybe the early onset of some of the diseases, maybe taking a geroprotective combination regimen is more beneficial than not taking it, even despite of the adverse effects. So that needs to be seen. And we developed a software system called Geroscope, where you can play around with pathways, create your own database of pathways. So here for my profile, I have 117 that we're looking at. Every pathway is annotated, so for every element you have the activator or repressor role, and it's manually curated. You can edit the pathways, group it, visualize it, play around with it, and run multiple experiments at the same time. So I'm just going to skip through it. So for some of the samples, you can calculate the keys to norm for every individual element in the pathway across a variety of samples. And you have various calculation parameters that you can set. So whether you want to choose the calculation algorithm on geometric or arithmetic values, what kind of sigma filter would you like to set, and what kind of Gero score would you like to get. So we have several. Again, looking at drugs that minimize everything between one state and another state, or drugs that minimize just the pathologic pathways. And so Geroscope, we started using it for drug discovery, but in the future we believe that it probably would be possible to use it for personalized medicine, clinical trial enrollments, and experimental medicine. So that's what we're using it for mostly ourselves. And this is our team at Hopkins, so I'd like to thank my team. This is our team globally. We have 33 people, mostly bioinformaticians. Scientist Advisory Board. So Charles Cantor contributed the most. He was the ex-director of the Human Genome Project. And if you'd like to collaborate in any way, we would like to work with you. Maybe take some gene expression data that you would like to process in humans, norm versus disease, and see what drugs work on your data set. Or if you would like to explore your area even further, we could develop a software product that targets your specific condition, and in most cases we won't charge you for them. Thank you very much. Let's collaborate.
All right, thank you. Do we need questions? Anybody? No. One question, yes.
So I was wondering, in your comparison between, for instance, peripheral blood samples from young versus old people, do you take into the equation that the cellular compartments actually and the composition changes over time dramatically, and how can you correct for that in your studies?
Right. So with hematologic samples, our software does not work very well. So we work mostly on solid tissues. But with hematology, we could try to look at a specific cell type. So after we collect a specific cell type and try to account for that variation with age. Then we would try to make those predictions. The paper on leukemia that we published, it was just one age of the patients. And yes, there was variability in cell counts, but we still managed to find common patterns in gene expression for a specific condition. But yes, I agree that there is a huge variability in terms of cell counts, and for hematology, it's very difficult to normalize. And there we would need to have significant human interaction, so to understand what's going on the patient level before we can make any predictions.